Good afternoon, everybody. I just wanted to welcome back Jim McLaren for the second part of his Genetics 23andU program. Thank you, Jim.
Thank you. Yes, thank all of you who came out again in lousy weather this time. Appreciate it. I said last week I had a video I wanted to show you, and let me get it set up here. And I want to show you what cell division looks like. And remember, every time cells divide, and it's going on millions and millions of times in your body right now, every second. Every time it divides, it has to copy all of its genetic information so that one copy can go to each new cell. What you see here, this is a plant cell, and it's already sort of well into the process of excuse me, of dividing. And what you see is there was a membrane here, the nuclear membrane. It kind of dissolves and disappears. It's kind of Mr. Wizard magic show. And then these things begin to coalesce, seemingly out of nothing. This is DNA, basically. These are chromosomes. And the DNA is coiling and coiling on top of coils, so that it becomes short and thick, and therefore easier to handle. So we'll continue the process here. Where am I? There I am. They are arranging themselves. There are fibers, which you can't see here, which are locating kind of a midpoint on each chromosome and pulling them so that they line up in the center of the cell. Boom. Whoa. There they go. Okay? And now, watch them just kind of fade out. This is a new cell wall separating the two cells. Okay? This is an animal cell, very briefly, highlighting those fibers. But I want to move on to the next guy. This one is an animal cell, and I'll just stop it briefly. And there you saw more of the sudden appearance of the chromosomes. And as the process goes on, I want to try to show you a, oops. Pause, will you? Kind of lost him. Oh yeah. See this guy right there? That's one of the chromosomes. He's misbehaving. He belongs over here. Okay? So, but just keep a little eye on it. There he is. Everybody else, all paying attention. Okay? Hey, get over here. Where you belong. Okay? Same thing. There they go. And in this case, the cell pinches off. Animal cells do that. So.
Imagine being able to build an app with your name on it.
No,
just
don't.
By thinking about it. Meet Base 44.
Okay. The next thing I wanted to show you, I want to show you duplication at the level of DNA. We're going to magnify now way beyond what you just saw. And, oh, I don't want you. I'm sorry. I tried to put all this stuff in the presentation, but I'm not technically smart enough. And this is the guy.
Using computer animation based on molecular...
I'm going to pause briefly. Get to full screen. Okay, she's going to talk your way through it when I start it. But here you're seeing DNA replication as we think it happens. This is an animation. This blew me away the first time I saw it. It was amazing. Anyway, this is the double helix that is going to be copied, okay? This guy right here, these are all proteins, all of these things. This guy right here, his job is to unwind the two strands of the double helix. And one of which you can see right here, okay? The other one is kind of buried a little bit, but you'll see it later. And it's coming out here, and it's doing this absolutely insane looping process. There's a reason for that. I don't want to get into the details because it would take the rest of the hour, and you'd be asleep. But I really just want you to watch it. And it's pretty close to full speed. I told you 2,000 bases per second. I think this is a little slower, but...
Research. We are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Okay. That just blows me away. I don't know if it does you, but the fact that we know what goes on at that level of detail is just, to me, astounding. So let's get back to where we were, which was, so we talked about Watson and Crick and how the DNA duplicates. And then we talked about how DNA is read. The language, that four-letter ACGT language, has to be translated into a 20-letter alphabet of amino acids to make protein. And this is the dictionary that I've showed you first, or right at the end, I think. And turns out DNA is read in what we call triplets, or codons, of 3 bases, okay? There are no spaces. It's not like English where you type a word and then hit space, okay? So there are no spaces, but the reading molecule, the messenger RNA, knows exactly where to start and read 3 bases at a time. And there are 64 combinations of 4 letters, AAC, AAC, AAT, AAG, etc. That's just 4 times 4 times 4. Every one of them means something, okay? There are no missing pieces here. And the fact that there are multiple words that mean the same thing, okay, synonyms, if you will, okay, any of those will put this amino acid into a protein at that location, and in these two included. And that's interesting, because if you start messing around, if you have a random mutation in DNA, that means the wrong base gets stuck in there somehow. Well, lo and behold, if you happen to change the last letter of that triplet, you're still going to put the correct amino acid into the protein. Very nice. It also turns out that if you mess around with the middle letter, you will probably, most likely, substitute a different amino acid, but one with similar properties. So it's less likely to upset the final protein if you have two pretty similar amino acids there. You mess with this guy, and you got problems. The first one. You're almost certainly going to change the protein. And, excuse me. And I also mentioned this, I think very briefly, but the business of mutation, which I just described, and the universality of the genetic code. Every living thing on planet Earth uses the same words to mean the same amino acids. That, to me, is the one strongest piece of evidence that you and I and those trees out there and the earthworms in the ground have a common ancestor. Okay? I'm going to go back 3 billion years to and more than that actually, to the first living thing. Okay? Darwin called it that mystery of mysteries, the origin of the first living thing. He just threw up his hands on that one. He said, give me that one and I'll tell you how you got here. But that's been the subject of a lot of research in the last 50 years. And we've actually got some pretty good ideas of how that first thing came about. But that's a whole different story. So here are some of the kinds of mutation that can occur. Here's a protein, okay, that's got this particular sequence of amino acids. And remember, the shape of the protein is determined by that sequence. Some attract, some repel, some like water, some don't like water, okay? And they'll twist and coil that chain into a three-dimensional shape. So here's the normal sequence, okay? And here's what's called a silent mutation. If at this position, at that position, that T, gets substituted for the A here, GTA says valine, okay? Well, over here, we changed the third position to, instead of GTA, it's GTT. Well, if you go back to the dictionary, it says valine. So it's called a silent mutation. Nothing changes. Missense, okay? Which one are we going to futz with? This guy right here, okay? CCC proline gets changed to ACC. Okay, we missed that first letter, and it's a totally different amino acid. So that thing is going to maybe push the chain in a different direction, fold it so that the final shape of the protein is different. And shape is everything. Shape determines function. Okay? And here's bad news. Real bad news. If we mess with this last one, okay? If we change TAC, which is for this amino acid, to TAG, it's called a stop codon. And that says to the transcriber, the molecule that's being made, that says, stop reading here, okay? I said there are no spaces, but there are 3 words, if we back up. Where are they? Those 2 guys right here. We got U's here instead of T's. Don't worry about it. There's a good reason for that. But these 2 triplets tell the reading machinery, stop here, that's the end of the protein. That's the last amino acid in the protein. And there are 3 of those, these 2 and this one. And then down here, this says start the protein, okay? So that's how the reading system knows where one protein starts and ends and the next one begins. It's really brilliant. What can you say? Why am I not advancing here? And here's a very specific example. There's a disease called sickle cell anemia. Sickle cell anemia is nasty. It was discovered in World War II. It's much more common in blacks than in any other population. And there's a really interesting reason for that. But they have a form of hemoglobin, the oxygen carrying protein in your blood. They have a form of it which doesn't function as well as regular hemoglobin, but you don't notice it all the time. It only affects you at certain times. And those times happen to be, if you travel to Denver, where the air is thinner, less oxygen. The hemoglobin molecules, because they have a slightly different shape, start clumping in strange ways. And they make your red blood cells take on these sickle shapes. And when you try to send those through blood capillaries, they grab onto, they catch on each other. And so they will block the blood flow in these microscopic capillaries and starve your cells of oxygen. It's not necessarily lethal at the time, but if it keeps happening over and over again and is particularly severe, it will kill you. It was actually discovered in World War II, prior to World War II, when Mussolini was bombing Ethiopia. The British took Ethiopian pilots to England for training. And they noticed when these guys were at 20,000 feet, they started fainting. And that's where this disease was discovered. It's very common in certain African populations. 10% of the population carries this gene. They're okay. They survive okay. But when you get the 2 genes together, in about 3% of the population, those kids die without good medical care. If you overlay a map of the prevalence of sickle cell, a map of Africa, with the prevalence of malaria, it turns out that having that gene, one of them, makes you much more resistant to malaria. So here's a gene which seems to be harmful, but in other circumstances, if there's malaria around, it's quite useful. But the key is hemoglobin, the part of hemoglobin that is affected is about 150 amino acids long. These are the first 6. First amino acid in normal hemoglobin is valine, followed by histidine, leucine, threonine, proline. Okay? And most of the time, you have a glutamic acid in this position, position 6, and another one in position 7. But here's the mutation in sickle cell. It's instead of TA, it's AT. Okay, and a valine gets put in there, and the hemoglobin molecule, instead of bending this way, the first 6 of the 130 amino acids just bends a slightly different way, slightly different shape change, and this is what happens. So the business of DNA to protein is really what it's all about. This is a quotation. I can't quote it exactly, but I heard it. I was sitting in class in college, and the professor in that class, he wasn't a Nobel winner, but he could have been. He said that to me and the rest of the class. Okay? And I've never forgotten it, because it was the first time I learned never say never. Okay? This guy, his name is Frederick Sanger. Frederick Sanger, in the early 50s, I believe. Okay? Won a Nobel, 1958, for determining the amino acid sequence of a protein. Valine, threonine, arginine, what I just showed you. He did it for for bovine insulin, which you could get in large quantities because diabetics were using it. It's a very short protein. It's one of the shortest proteins in your body. It's only about 50 amino acids long. And he determined the sequence. Took his lab 2 years to do it. It was backbreaking work. Lots of PhDs were minted in that lab from that work. We can now sequence proteins 3 times longer than that in an hour. And there's a machine about the size of this projector. And you put the purified protein in a container over here, and a little tape comes out the other end. Says, here's the sequence. Okay. So Sanger wins his Nobel Prize. Everybody's totally impressed. And along about 1980, I was in that class in 65, I believe. Along about 1980, same guy, okay, says, you're right, the method I used for proteins, that won't work with DNA. I got to come up with another method, which he did. It was brilliant. And he got his second Nobel Prize. Amazing. Most of you know that we have now, not only can we never sequence DNA, we have now sequenced the entire human genome. First draft was proposed with lots of blank spaces and question marks, but in 2001. And then they cleaned up the blank spaces, okay, figured out what should have been there, and the full sequence in 2003. 3 billion base pairs, 3.2, but what's 100 million plus or minus? You could store that on 200 gigabytes of computer memory, but it wouldn't be very useful. You have to put a lot of extra data about which chromosome this is on, a lot of stuff. So actually it's a few terabytes, but manageable. Searching it is a whole other story. We'll get to a little bit. The initial cost of that process, huge consortium of people from all around the world, was about 2.7 billion. That's spread over a number of years. You could now send a little swab from your cheek to, well, not 23andMe anymore, but to wherever, I don't know where you'd send it. But you would get back your 3.2 billion base pairs for about $300. Okay. It's been automated to that extent. It's just astonishing. Who did that? These two guys were primarily responsible. There are hundreds of people that were involved in this in bigger or smaller ways. Francis Collins of NIH took over as in charge of the international consortium, and and Craig Venter, he's an interesting guy. You should read about him sometime. He founded Celera Genomics, and he wanted to do this privately and basically patent it. But he was finally convinced that that wasn't a good idea. So he had a lot of data to combine with the other data, and finally the genome was printed out. So a little bit about the early findings. The biggest surprise from the whole thing, 3.2 billion base pairs, okay, giga base pairs. Only 20,000 genes that make protein are found in that mass of DNA. You say, well, that's a lot of genes. No, it's not. It is, of the 3.2 billion, protein coding genes occupy 20%, roughly. That's it. All the rest is this stuff. It was originally referred to as junk DNA. It was a very bad word to select. It just meant I'm stupid. I don't know what it is. Okay, all that other stuff. If you look at the genome of E. coli, the bacteria, or pretty much any other bacteria, there's very little junk. It's all coding. It's very efficient. So the real question becomes, what the heck are you carrying all this other stuff around for? That requires a lot of energy on the part of a cell to replicate stuff that's useless, that's junk. Well, we've identified a lot of things, and I'm not going to go into each of them. There's still 9% that we just throw up our hands and say, I don't know. That's a lot. And probably because we just don't know enough yet and haven't sorted that out. But here, pseudogenes, that's a really interesting group. This is a group of genes. Genes like to copy themselves and go somewhere else and park on a different chromosome maybe, or somewhere else on the same chromosome. They're called transposons, the ones that do that. And when you copy a gene, say for hemoglobin, and you have 2 copies in the gene in your genome, one of them can mutate independently of the other one. And sometimes you get different functions. You've got about 8 or 10 copies of hemoglobin. One of them is called fetal hemoglobin. When the fetus is growing in the mother, he's competing for oxygen at the placenta. Fetal hemoglobin, when the mother's hemoglobin goes by carrying oxygen, fetal hemoglobin just says, give me that, okay? I got a stronger affinity for oxygen than you do because I need it, okay? I'm growing. After birth, the fetal hemoglobin gene gets turned off. So, and there are a bunch of other, but there are some genes that are very similar to hemoglobin, but so different that they have no function. And they're called pseudogenes. So you got a ton of those hanging around. And then you got this huge defective transposons, functional transposons. Transposons are just chunks of DNA that decide, I don't like it here, I'm going somewhere else. And they get plunked down on a different chromosome, just wherever they happen to fall. Fortunately, you've got all this DNA which isn't doing much, so 99.9% of the time, nothing changes. But if that otherwise functionless stretch of DNA has no function that you can identify, but if it gets stuck in between other genes, it can affect how they are controlled. So things get really complicated real fast, okay? But I just think this is interesting. So I thought junk DNA to me is just a bad term to have chosen. Okay, you might wonder whose genome it was. Was it one person that they picked? There was some talk that it was Craig Venter's, and I bet you that a lot of it, of the original draft, was Craig Venter's, but they chose about 17 different people, 18, 20, I don't know exact number, and it was kind of a mishmash of them. And they would do them separately, line them up, and say, well, in 15 of the people, it's a G here at this position. In 2 of them, it's something else. So we'll kind of take an average, okay? Since then, things have gotten much more precise. So if you were to send your DNA in for a full, they don't do full for everybody, okay? But what we find is that SNPs are a major part of what makes you unique, unlike anybody else on the planet. SNPs, single nucleotide polymorphisms. That's why we call them SNPs. In a given gene, okay, or a stretch of DNA, one base may be different between you and the mass of people out there, okay? I've got one of those in the gene for one of the blood clotting proteins. It's called G20210A. I don't remember the exact number. It means I have a G at the 20,210th spot of this protein. And it makes me a little more likely to have DVTs, blood clots in my legs. So I'm careful about that. I only know that because my daughter had a DVT and we got genetically checked, and it was my fault. I gave it to her, and I did to my other daughter as well. But it's these little unique single base, okay, G instead of an A here, so on, that are not totally responsible, largely responsible for what makes you different from any other person. There are other issues involved. Okay, got a genome. What are we going to do with it? Because if you look at it, and you could look at it, if you get on NIH website, there's a big learning curve on this thing, but you can access the human genome at home on your computer. I've done it. I looked up that G20,000, whatever, and it was right there. So you got this big, huge database. It has to be, it's all ACT, line after line after line after line after line. So it has to be computer accessed. There's a whole field called bioinformatics. And all you do is sit at the computer and look at sequences and compare sequences, okay? It's more than that, but, well, let's return for a bit. Let's go back in time to the 1980s. There were discoveries made in the 1980s that blew people out of the water. And they were based on that observation that the genetic code is universal. You say, well, if it's universal, maybe I could take a unicorn gene and stick it into a bacteria, right? There's a bacteria, and maybe we can get it to grow a horn, right? That's a little silly, mostly because nobody could find a unicorn. If there were such things, somebody would have tried it. But people in labs made discoveries that allowed us to think about engineering the genes. Taking genes from one organism, put it in another one. Because if I put that unicorn gene into a bacterium, it's going to make that protein. It's just DNA. It's just A's and C's and T's and D's. And so the machinery of the bacteria will happily read whatever you put in there and make that protein. But the scientists didn't invent this stuff. They found it in bacteria, the ability to do this in bacteria. And we've only borrowed their ability to manipulate DNA that they've taken billions of years for them to evolve. And the first major discovery was of something called a restriction enzyme. It's a protein, right? Enzymes, proteins. E. coli has this enzyme that just spends its time crawling over DNA, base after base after base. And if it finds this sequence, GAATTC, anywhere in the DNA, it goes, cuts it. It cuts it in a special way. It cuts it here on this strand and here on this strand, okay? So you got this, we call them sticky ends, okay? Down here, you see. there's this exposed AATT and an exposed AATT. Okay? Well, what do exposed bases like to do? They like to find each other. They like to form these pairs, right? So, the other thing to notice, weirdly, and I've never heard anybody explain this, there are a bunch of different restriction enzymes. They all recognize, almost all of them, recognize sequences that are palindromes. They say the same thing forward and backward. Okay? GAA-TTC, GAA-TTC. Weird. This palindrome is 6 bases long. Others are longer, others a little bit shorter. Okay? So this protein came to be called EcoR1, E. coli restriction enzyme number 1, first one to be discovered. And they all behave in similar ways. Different species have different restriction enzymes, which is a good thing. Here's a list. It only goes from A and gets a few of the B's, okay? And it goes on through all the bacteria species. There are hundreds of these things. But if you look at them, these are the bacteria that make the enzyme. These are the guys that have discovered. And if you look at how they cut, GTC, GAC. Well, it would be GTC, GAC. Palindrome, okay? Most of these cut to produce sticky ends, okay? Some of them require very long sequences, and you can have anything in between here, and it will cut. So people say, oh, scissors. I've now got scissors to cut a gene out of, say, a human cell, stick it into the bacteria, use the scissors to cut the bacteria DNA, and use those sticky ends to give you a full, I'll show you an example here in a sec. It's brilliant. Before going on, you say, well, why are bacteria got these enzymes that will cut DNA? You got this 6 million base pairs chromosome that the cell uses to reproduce, okay? That little 6 base sequence, that's got to occur many, many, many times. So why do you have an enzyme that's going to chop up your DNA? Well, number one, you minimize the number of that sequence in your, and number two, you chemically modify the nucleotides in that little short sequence so that the restriction enzyme can't cut your DNA, okay? Bacteria have this interesting habit of carrying what are called plasmids. Plasmids are short loops of DNA. 1,000 bases, 10,000, okay? That's very short. And the two ends stick together to form a loop. These things are used by the bacteria. They can be passed from one bacteria to another. And these are where antibiotic resistance come from. These plasmids carry resistance to not penicillin, but erythromycin, okay? There's a gene in a plasmid that makes the whole cell resistant to that antibiotic. So that cell can reproduce. If you don't have that plasmid, you don't reproduce. But you can pass it to your buddies, okay? Well, that's all interesting, and doctors care about it because of antibiotic resistance. But But the genetic engineer said, oh, cool. I can use these things to transmit a gene. I will take plasmids out of the cell. I'll cut them with the restriction enzyme. It's got sticky ends, so all of a sudden it's not a loop. I'll put my gene that I want from some other species in the mix, and occasionally I'm going to get the plasmid with one sticky end and the other one to include the gene I want. Well, interestingly, this set everybody's hair went on fire, okay? Oh my God, I can do this, I could do that, I could do okay? Just imagine. Anything you could imagine, you could say, maybe we can do that. Well, this guy, Paul Berg, convened a conference at Asilomar, California. It's this lovely place overlooking the ocean. He was a professor of mine, actually. In 1975, he said, slow down, put the brakes on. This is scary stuff. Let's get together as many people as we can and see if we can agree on some restrictions on this technology. They really wanted to put a moratorium. They wanted to just stop this research for a significant length of time, just to see if we could get agreement. And it was actually surprisingly successful, totally voluntary. Nobody could make any laws. But it was from that point on that genetic engineering proceeded with real safety safeguards. You can imagine scenarios where you put a gene for, I don't know, some disease into a bacteria and then let it out of the lab. You know, hair on fire. Well, this is perhaps the first, I think it is the first really practical use of genetic engineering. Here's in your genome somewhere on some chromosome. I'd have to go look it up. I don't know which one it's on, but you could look it up in the genome database. And here's the human insulin gene. Okay, among those billions of base pairs, this one is about 300 bases long. Okay, so you just take human cells, purify their DNA, expose them to EcoR1, a restriction enzyme, and it cuts the human DNA into millions of little fragments. You've got to find the fragment that has the gene in it. There are ways to do that. Trust me. I'm going to take a plasmid, this little thing. You can order these, okay? There's catalogs, and you know, I want 50 milligrams of this plasmid. There are thousands of them you can choose from. So you choose one that will respond to the restriction enzyme that you want to use. You just cut it and open it, okay, into a linear molecule. You mix the two, the DNA with the insulin gene with the plasmid, and you hope that the sticky ends, one there and one there, find each other. You use a little enzyme called ligase to connect the bonds permanently. Now we take this plasmid that we have engineered. Oh, shoot. Oh, wrong button. Okay, we take this plasmid, we expose the bacteria to it. Bacteria, remember, they exchange plasmids, so they have mechanisms for taking up the plasmid as part of themselves. Now they reproduce, put them in a big fermenting tank, stainless steel, 500-gallon fermenting tank. Plenty of food, the right temperature. They multiply into massive numbers. You harvest the bacteria, you purify, go through a bunch of purification steps, and lo and behold, you have purified human insulin available now to diabetics who don't respond well to cow or pig insulin, which had been used for a century. Now can take human insulin, engineered human insulin. There are many proteins that have gone through this process. Mentioned 3 of them. Factor VIII is one of the proteins involved in blood clotting, and it's the one that's missing in hemophilics. Hemophilics through the 80s and part of the 90s could get treatment by going in and getting injections of factor VIII protein. That protein was purified from a single dose, would have been purified from 50 pints of human blood, donated human blood, to extract and purify enough factor VIII for 1 injection. Hemophilics needed about 1 injection a month, so the cost was astronomical. And the other cost was you had a very high chance of dying from AIDS, because those transfusions supplying those 50 pints a month were coming from the general population. And they were coming from blood banks that paid people to donate. And at that time, there was no screening process for the AIDS virus. So a number of people, and we know one individual, hemophilic, and died of AIDS. By the mid-90s, we were producing factor VIII from bacteria by that very method that I just went through. It's purified, it's human, okay, and it's relatively cheap. FSH, follicle stimulating hormone, that's one of the hormones involved in stimulating human egg production in the monthly cycle. So for women who don't produce it or produce a defective form, very useful. And good old HGH, human growth hormone, okay? It's a short protein, and it is used to treat people who don't make it, who would otherwise be dwarfs. But unfortunately, it sort of got into the general market, and somehow athletes decided, oh, this would be a good idea. I've never understood what it did for them, okay? Maybe it had a positive effect on their performance. Mostly they were taking steroid hormones, but HGH as well. But again, a very useful hormone in the right circumstances, but like everything else, abuses can follow. So here comes genetically modified food, okay? Instead of putting the protein into a bacteria, let's put it into a tomato, okay? Let's take the gene rice. Let's take daffodils that produce beta carotene, the enzymes that make beta carotene, vitamin A, okay? And let's stick it into the rice plant. So the rice plant makes vitamin A. And golden rice, okay, is taken off, is very useful, particularly in areas where vitamin A is not readily available in the diet. That's one I think is a good idea. Flavor saver tomatoes, remove a couple of genes so that it won't spoil as quickly. This one scares me a little bit. Kids used to ask me all the time, what do you think of GMOs? You know, hate GMOs. GMOs are terrible. And my basic response was, we've been doing genetic engineering for 100,000 years by artificial selection of the animals and plants that we grow on farms. It's just slow and inefficient. So if you put a gene in another organism, it's made of A's and C's and T's and G's. And when you eat food, you digest the DNA in food into A's and T's and C's and G's, and then use those to make your own DNA. Excuse me. So to me, what's the big deal? I don't have a problem with it. Except in certain cases, BT corn modified with a bacterial insecticide, okay? So the corn plant itself makes this insecticide. It may be perfectly harmless, okay? It's a protein. And you digest proteins when you eat them too. But it kills bugs. And if it kills bugs, you know, maybe I have a problem with that. The other problem I have with genetically modified corn is Monsanto Corporation. Pardon me if any of you ever worked for Monsanto. But they produce a genetically modified corn that they purposely sterilize. So you cannot use the corn that you harvest as seed corn for your next year. So you got to buy it from Monsanto. Okay? Thanks a lot, guys. Really appreciate that. Anyway, salmon modified with the growth hormone, they grow faster. Again, I wouldn't have a problem with that myself, but. Oh, and this one, glow in the dark animals. Okay. This was kind of where they said, oh, let's take a unicorn gene and, you know, put it in a human. I don't know. Just to see if you could, okay? And you can. And there are rabbits and other species that they've done it to. I don't know why. And it raises this issue as well, okay? Once you have a genome, we don't have just the human genome. We have now genomes in computer banks for thousands of species of animals, of plants, of fungi, of protozoans, of bacteria. And you can all look this stuff up. It's all, I could go find the genome of, I don't know, an earthworm on my computer. And it would be A's and C's and T's and G's, right? So being able to do this genome stuff, there are companies and labs that are working on this, okay? In order to do it, you have to get the DNA from the extinct species, okay? And if you're thinking Jurassic Park right now, you can stop thinking Jurassic Park, because the probability of getting functional, useful DNA out of a 60-million-year-old rock is pretty near zero. Never say never, Jim. But the science in that movie was bad is an understatement. It was a great movie. I loved it, but what can you say? These are just some of the animals that we think about. Maybe we bring them back. Dodos, there are enough specimens in museums from which you can extract DNA, okay? Enough that maybe you'll be able to do this. The Tasmanian tiger, I don't know why it's called a tiger, it's a dog, basically. The stripes, I think. And of course the woolly mammoth, okay? These guys get frozen. 30,000 years old, but they freeze. And from that material, you can extract some pretty good DNA, some pretty good genetic stuff. So the thought is we'll take the genes that we can and we will put them in an elephant egg that we've destroyed the nucleus, and that egg will be put into a foster mother and raised, and maybe we can make a mammoth, okay? You probably read a couple months ago, 4 or 5 months ago, the company that has brought back the dire wolf. That's an overstatement, okay? They put some genes from dire wolf into an actual wolf and brought back some of the characteristics, but it's not the original, okay? And I find this stuff kind of interesting. I don't know if I think it's worth all the money, because there are a lot of ethical questions. What are you going to do with a mammoth? You can put him back out in the wild, eating plants it's never seen before, you know, interacting with animals, okay? So a lot of questions about it. And how about humans? Neanderthals. This was a scientific tour de force of extracting human DNA from bone. You go get Neanderthal bone and you drill in and you get enough DNA. It's all in fragments, it's all in pieces, but you get enough that you can overlap and see what's different in modern human, from the modern human genome. And you find enough unique sequences that say this is Neanderthal, this is not human, okay, or modern human. Neanderthals were human, no question about it. It's very difficult to isolate, okay, the guy who worked out the techniques, because when you get DNA out of a bone, there could be bacterial DNA in there. There could be bacteria from the plants that were sitting on the bone for 30,000 years, decaying along with it. So it was not easy, but it's been done. And Merry Christmas, guys. You've all got some in your genome, okay? Some of it was unique to the Neanderthal genome and is found in modern humans, not in Africa, interestingly. We know Africa is the cradle of humanity. All the early Australopithecines and Lucy, it all started there and all migrated northward from there. Neanderthal, the oldest ones appear about, I think 200,000 years, and they migrate up into Europe and over this way, okay? They were very common. But there'd always been the controversy of modern human, Cro-Magnon. Did they mate with Neanderthal and kind of wash out the genes, or did they just kill them? And my tendency was to think they probably killed them. I mean, that's humanity. history. But we've still got enough of their DNA in our genomes to think, you know, maybe it was both, or, you know, combination of both. But you and I have up to 1% probably for us. If you go over into Asian populations, the percent goes up to 3 or 4. Okay. It's just, in it, you know, gee, we've got a genome. What are we going to do with it? You can do all kinds of interesting stuff. Okay. And again, varies from less than 1 to 3% throughout the world. And this will bring us, how are we doing here? Ooh, I've way overshot. I'm sorry. Gene therapy, okay? Could we cure a genetic disease? It's a problem, because if you have diabetes, if you have, name a disease, Huntington's disease, it's in every cell in your body. And if I'm going to transplant a gene, how am I going to get it into every cell in your body? So that's been a huge issue with this process. Germ cell, if you do it with an egg cell, or 2 or 3 cells, 4 cells, okay, then you have a chance of curing a fetus, an embryo, at that stage. If you could do it there, that would work. But for you and me, it's a little late. How do you get the genes in? Okay, 2 major methods, but one is to use a virus. Okay, you use a virus particle. They're really good at attaching to cells and injecting their DNA into you. That's how they work. So what you do is you take an adenovirus, which is actually a harmful virus, but you destroy the genes that make it harmful. You insert the gene that you have engineered into the virus. The virus attaches to your cell, injects that DNA, and you hope it's incorporated into the nucleus. This has worked well enough. You can also use things called microseals, little, well, never mind. But you can do it, and it has recently been used with sickle cell people. Because sickle cell people, the only place that's having a problem is in the bone marrow, the red cell producing cells. And if you can get the gene into enough of those cells, they're having some progress there. But so far, that's about it. So, and you've heard of this, okay? That's what it means, okay? And it would take me a week and a half to describe it to you, okay? And you would go out and sit and forget it in about 5 minutes, because that's what I've done. I've read it, understood it, and 5 minutes later I couldn't explain it, okay? But what it does, unlike a restriction enzyme that's sort of random in it where it cuts, just depends on where those sequences are. CRISPR, you can go right to the G that you want, cut the DNA, and stick an A in there, okay? Highly targeted. It's exploding, research in this area. So that's it. Thank you. I'm sorry I did overshoot by a lot.
Thank you.
Thank you.
You should raise your hand and say, the bell rang.
Let's be on the 10th.
Yeah.